Good day everyone. We're still continuing our discussion of GIT infections. In current lecture we'll talk about one of the most dangerous microorganisms, which is known as the Vibrio cholera. In the contrast with other pathogens discussed in previous lectures, it's not enterobacteria as a lot of students think. It is separate family Vibrionaceae and separate genus Vibrio. Actually, we have few additional species which you can meet in clinical practice, but of course the major pathogen is Vibrio cholera. So as well, it is gram-negative rod-shaped bacteria. It can have some curved shape or some resembles comma shape. It has single polar flagella, which gives unique shape to that bacteria. By type of respiration, it is facultative anaerobic, means it is both aerobic and anaerobic bacteria. Vibrio is very sensitive to some environmental factors. It prefers alkaline media, so it is very sensitive to acid, and people who have low acidity in stomach are more affected by diseases caused by Vibrio. Dry and exposure to direct sunlight are bactericidal for Vibrio, so bacteria cannot survive a long time period without water in open area. And naturally, Vibrio is aquatic organism, and a lot of species are natural aquatic organisms. Vibrio cholera is not an exception, and only human is susceptible to the disease, and Vibrio cholera diseases are described only in human. So bacteria is famous and known because of pandemics it caused in past history, and a lot of people, millions of people have died. And actually for your faculty, that bacteria is the most important, and we will ask and examine that microbe in detailed manner, because 7 cholera pandemics described in human history all started in India in Ganges river, that Kumbh Mela festival, when a lot of people swimming in water, they are drinking and washing with that river water, and disease is easily bacteria is easily infection in human. Then those people goes home, they had a lot of contacts, they go into other parts of the country, and disease is easily spreading by human to human contacts through low hygienic conditions, through fecal oral route or other. And recently 2 outbreaks, they are not even pandemics, but very important outbreaks were registered. One previous century South America, and recently in Yemen. That disease, that outbreak is not finished. Some accidental cases are still registered, but a lot of people suffered because of low supply of pure drinking water. So bacteria, of course, was suspected to be used as biological warfare, because easy to deliver to human and infection dose is low. Everyone susceptible to cholera, and disease have high mortality rate without proper treatment and without proper water supply. So bacteria needs ordinary media, but addition of salt, low concentration about 5%, and alkaline pH are preferred to grow that bacteria. Of course, initially you will need to differentiate with enterobacteria and with the pathogens of Enterobacteriaceae family, and the same culture medium Acontia agar is required to grow that microbe initially. Colonies will be lactose negative, very small, pinpoint, and pale pink. Second culture media which is required for cultivation is a blood agar. Usually it is a sheep blood agar, and colonies with hemolytic zone around required to be studied with the oxidase test we discussed before, and if color is blue, color of colony or color of sample, you will add that oxidase reagent is blue, then it is confirmation of pathogenic Vibrio species. We have alkaline peptone water for growing of bacteria. It is enrichment culture medium, or if you need to take sample and deliver it to lab, of course again humidity is required and unique pH is required. That culture media is suitable as a transport culture media, but laboratory with specific diagnosis should use TCBS agar. It is agar which allows to grow Vibrio and suppress other bacteria from intestine and environment, and it helps to differentiate Vibrio cholera and other Vibrios such as Vibrio parahemolyticus. So initial culture is green. If Vibrio cholera will grow, you will have yellow colonies, and agar around colonies will change the color into yellow as well, while other Vibrios such as Vibrio parahemolyticus will remain blue or transparent without any changes in agar, and you can identify that extremely dangerous bacteria even by growing and culture media without some additional tests. But of course, it is required to know, is it pathogenic and epidemic Vibrio cholera or not. And the very first classification and the most important classification which is used for differentiation of Vibrio cholera, it is a serological classification according to structure of somatic O antigen or lipopolysaccharide. And then nowadays Vibrio cholera is classified into 139 serogroups. And all strains caused pandemics in history belong to O1 group. So of course it is much easier because the only group need to be identified. We have agglutinating serum, it is called O1 serum, and if you perform test and test is positive, then it is pandemic emergency. The rest bacteria, the rest strains are called as non-O1, and it's not really important which one serotype you have. While it is non-O1, then pandemic is not possible and it is only a local problem. So it was really so until recently, about some 30 years ago, new strain O139 was described. It was identified in Bangladesh and it caused some outbreaks in history, and it was confirmed that actually not only O1 but that strain can cause epidemic as well. Symptoms are identical, properties are identical, but of course it is another serotype, and O1 sera not working in that case. And actually nowadays to identify pathogenic pandemic strains, you need to use two types of serum, O1 and O139. The rest bacteria can only cause vibriosis, and cholera is not really possible in massive manner, so it's not really so dangerous. Second classification is according to biological properties. Vibrio cholera O1 group is classified into two biotypes. It is a classic biotype which caused epidemics and pandemics at the very beginning of the history, and new mutated strain L-tor, actually it is the same Vibrio cholera but with some mutation, and differentiation is possible by studying of bacterial growth on blood agar. L-tor is actively hemolytic, while classical show you only small zone of hemolysis. Diseases caused by L-tor strain are more aggressive, bacteria is more harder, more adapted to environmental conditions, resistant to some antibiotics, to polymyxin B for example, and it is used for differential diagnosis on petri dishes and lab. And in the case of L-tor, some cases of chronic carriage were described, so it is possible to have asymptomatic people who can spread disease in community. So according to structure of antigen, both biotypes, classic and L-tor, have one antigen which is common and found in any strain. It is antigen A. But one more antigen have variable structure in different groups, and it was identified in two modifications named B and C. And actually nowadays we have serotypes of O1 serogroup. It is Ogawa, Inaba, and Hikosima. If A and B antigen is present, it is Ogawa. If A and C antigen are present, it is Inaba serotype, and rarely we can have all three types of O antigen A, B, and C, and that strain was called, that serotype was called as Hikojima. Again, it is important in identification and in epidemiology, but in clinical practice symptoms are not really different. So bacteria, again, it is a gram-negative bacteria, have lipopolysaccharide in the cell wall, and the toxin is present, so all functions are applied. Bacteria have a number of factors required for adhesion to microvilli of the intestine. It is hemagglutinins which can help to fix not only to blood cells but to epithelial cells as well. Pili, a lot of adherence factors. Bacteria is motile, again flagella is present, and it helps not only to survive an environment. That factor is important in invasion into mucous layer of small intestine. Bacteria can penetrate easier with the help of that flagella. Bacteria produce a number of enzymes such as proteases and uraminidase. Again, their lytic enzymes helps to destroy mucin layer of epithelium, helps to destroy connective tissue, and then to invade deeper in the epithelial layer. And bacteria is not invasive by properties, it just need to fix and hide out of our aggressive factors and flora. And the main factor of pathogenicity is a cholera toxin, which is known as well as a CT or cholera gene, and it exists in two forms called CT1, CT2. And most often CT1 type of toxin is produced by classic cholera strain, whereas CT2 produced by main number of O139 strains and main number of L-Tor strains. But again, few L-Tor Vibrio cholera strains were described with CT1 toxin, so that property is not for sure, but still. So if you talk about cholera, the disease is one of, again, most known because of pandemics and millions of people died. Disease, as any other GIT disorder, transmitted by fecal oral route through contaminated water or even some aquatic animals. Disease is absolutely natural. Reservoir of infection initially environmental, and again, Ganges River is the natural home of Vibrio. It is fine to have it here, it is living here, because no one except humans suffering from the disease. And initially the source is only one, it is contaminated aquatic reservoirs. Of course, nowadays a lot of water sources are naturally contained that Vibrio, not only India, some other countries, but India is the biggest reservoir for that microbe. But when some person became infected, then persons became a source of infection. And initially one human can be infected in some aquatic reservoir, but it will spread, or he or she will spread that infection to other people. And again, carriage is possible, so nowadays people can act as a source of infection as well. Incubation period is very short, usually it is less than half the day, can be prolonged up to 2 days, but rarely. And main course, again, it depends on immunity on the subject, drinking abilities and amount of clear water. So it can be very short, about 2 days, and can last up to 7 days, but in previous in history description, a lot of people died within 3 days after disease started because of severe dehydration. So bacteria will secrete cholera gene or cholera toxin, which will start acute onset of nausea, vomiting with huge amount of vomiting masses, watery diarrhea which resembles rice water. It is exceed 1 liter in volume stool with multiple mucous components, and it resembles some white inclusions like you boiling the rice and then washing it. So several episodes per day are possible, and in general people will lose several liters of fluid, up to 10 liters of fluid per day. Of course, it will lead to very severe hypovolemic shock, insufficiency of blood circulation, all organs will suffer, including brains. And again, if clean water and clean supplies are not available, then up to 50% of people will die within a few days after the start of the disease because of severe dehydration. As it's obvious from the pathology of the disease, the main type of treatment is replacement of electrolytes and fluids. Bacteria is present only in intestine, it's not invading, so in main number of cases antimicrobial treatment is not required. If you give a sufficient amount of water and fluids, then people can recover themselves. But of course, no one will want to risk, and no one will want to try their chances, and antibiotics should be prescribed almost always. Tetracycline is the classic drug of choice, and Vibrio is still susceptible to tetracycline group of antibiotics. The same measures as for any other GIT disorder: proper sanitation, proper cooking, and supply of clean water. So pathogenesis of cholera is kind of easier and more simple than in case of Shigella, Salmonella, and other enteric pathogens. Bacteria is ingested with the water, and initially it needs to pass acid barrier of our stomach, and people with good acidity in stomach less susceptible to disease. Vibrio will die here very fast and very easier. But if acidity is decreased or Vibrio is ingested in high amount, then bacteria will pass stomach and get into intestine. It will fix to mucous layer of epithelial cells and start production of the labeled toxin. Again, it is biological toxin that have common structure. It is a protein with two main subunits, A and B, active for main effect and binding subunit made of 5 identical, or we call it pentamer. Toxin binds to receptor of epithelial cells and activates CMP enzyme, which causing release of excess amount of water and electrolytes, initially electrolytes and water in the intestine, and people will have watery diarrhea without blood and stool. So again, you have multiple confirmation and description of that disease in the human history. We have few very famous pictures which are present in different museums. One is the dead plain on femoral bone, when in Paris in France it was severe epidemic of cholera, and you see that a lot of citizens were dead. They were lying everywhere in squares in the streets. They just died during walking into some hospital looking for help, and they were absolutely dehydrated, and they were looking like a skeleton of the human because water was lost and bones were easily seen through that skin. And second one is, the second picture is presented in Vienna. It is 23 years old lady. She was visiting some painter just before the accident of cholera. She was absolutely healthy, and when that painter saw that woman a little later on the street, she already was infected with the Vibrio cholera, and just 1 hour after that infection, and you see that absolutely changed appearance. Woman became thinner, skin is pale and even blue because of cyanosis, and of course general appearance is awful. She's suffering, and in 4 hours after the infection she died. So not after infection, after first symptoms she died. So disease is really dangerous, really famous, and it's absolutely important to identify Vibrio cholera 01 or 01 39 serotype as soon as possible. And of course, if it is suspension on epidemic or even in pandemic, you will not have enough time to take sample, inoculate in culture medium, grow in agar, perform some tests. We need to do something right now. And we have rapid diagnostic dipstick tests. Again, multiple classification, multiple manufacturers are present. Crystal YC is approved by CDC. It is international test, but we have a lot of some looking like tests. The principle is the same. It is immunochromatography, and antibodies against 0139 and 01 Vibrio cholera are fixed to that strip. You need to take a sample, add to paper strip, mix with some reagents which are going with the kit, and if that strip of precipitation will appear according to place where it appears, you can have result. It can be negative, positive for 01, positive for 0139, or both strains. It's fast, and again you can perform it near the bed of the subject. You don't need laboratory, you don't need specific staff. Test is easy to perform by any doctor, even medical nurse, or even subject him or herself. Of course, it is essentially and very important to prevent the disease, and we have a number of vaccines against Vibrio cholera, but routinely those vaccines are not used. They are indicated for people who are going to travel in endemic area, for laboratory workers and doctors who are working with those people with that pathogen. And especially it is important when some epidemic will appear and surrounding countries you will have one country affected, and all surrounding countries should perform emergent vaccination against cholera to prevent spreading by a continent and other continents. So according to types of vaccine, again we have two important types of vaccine. It is a live and inactivated. A live vaccine is more dangerous. It is indicated only for adults 18 to 64 years old. And inactivated vaccine, again it can be oral. It is indicated for travelers and people 2 years and older, so it is more safe, and even kids can be vaccinated. We have a different schedule of vaccination for every vaccine according to instructions of manufacturer, but again any vaccine will give immunity at least for 2 years. some vaccine even 3 years, but some time period before vaccination and visiting the area is required. So if you need to be vaccinated, it's better to read instruction. And again, India is still hyperendemic by cholera, and if you're going to live in India, it's better to consult with the doctor. Maybe it is a point to vaccinate and to keep vaccination. In that case, revaccination every 2 or 3 years is required to maintain immunity. So one question difficult to identify, and you're always asking what is it? Vibriosis. We have a number of diseases which are caused by Vibrio, but not Vibrio cholera. The most known Vibrio after Vibrio cholera is Vibrio parahemolyticus, usually causing mild to moderate enteritis transmitted by the same elementary way, and raw fish, raw shellfish, and some sushi and other Chinese-like food, which is very popular nowadays, can act as a source of infection. The same bacteria is kind of sensitive to environmental factors, and proper cooking is a good way to prevent, but we have some cultures when fresh meat or fresh fish should be used. In that case, enteritis is very possible. Incubation period is less than a day, and usually symptoms will appear some 12 hours after ingestion of microbe, manifest by nausea, vomiting, and some common symptoms of GIT disorder, including watery diarrhea. In rare cases, can be with mucus and blood. Again, disease is self-limited. We have a lot of natural factors of protection, and mainly in few days, spontaneous convalescence will be observed, and the only therapy required is compensation of lost fluid, electrolytes, and proper diet on that period of that diarrhea. Second bacteria, which is more dangerous and more important, it is Vibrio vulnificans. That bacteria rarely causing gastroenteritis. More often it is associated with wound infections, and few cases of fetal septicemia were described. So in the case of enteritis, bacteria is transmitted by elementary way, of course, but if you talk about wound infections and septicemia state, then disease is transmitted by different cuts and injuries of the wound, which are obtained in the water, or if you already had a wound which is surgery or some other scratch, but it is deep wound, then swimming in the water or any contact with the water can lead to infection. Of course, group of risk is some fishermen and some professions which deal with aquatic activity, aquatic jobs, and disease is transmitted during some accidental trauma. Source of infection, of course, is the water. If it is some shellfish during cooking, people can accidentally cut and bacteria can invade into the wound. So in the case of gastroenteritis, the main symptom, of course, is watery diarrhea with some common symptoms. But in the case of wound infection, very soon after injury or after contact with the water, subject will feel common fever. In the place of injury, redness and pain will appear. It will rise, increase, swelling will grow. When edema will be kind of severe, discoloration of skin will appear, and discharge from the wound, aquatic liquid will goes out. So without proper treatment and without proper diagnosis, disease will progress to necrotizing fasciitis. We saw it during streptococcus lecture, and in the case of fetal septicemia, of course, systemic symptoms will appear, such as loss of blood pressure, critically high fever, and skin lesions near the wound will be blistering with production of massive exudate. In the case of gastroenteritis, replacement of lost fluid with water and electrolytes will be sufficient, but of course, if it is wound infection, immediately antibiotics are required in high doses, and if required, surgery. In very severe cases, when people not looking for medical help in first hours, that infection can grow, and even amputations can be the only way of treatment of the disease. In severe cases, even lethal outcomes are possible. And of course, again, non-specific measures of prevention are required. In the case of GIT disorder, it is proper cooking, proper sanitation. In the case of wound infection, if you have some open wound, recent, which is not healing, it's better to avoid any contact with the water, or if it is necessary, you can and you should close the wound with some bandage, use a medical glue or some other measures. But again, even if it is small wound, but it is obtained from some aquatic animal or water, it's much better to look for medical help as soon as any symptom will appear. Early treatment is the key to save extremities and save your health. So a few pictures of wound infection. In common literature and in common people, that disease is known as flesh-eating bacteria because actually it's very fast progressing and it's really dangerous disease and looking awful. Meat, muscles and skin are destroyed very fast. It will start with formation of some kind of bulla with necrotic discharge, and then only bones will remain, and of course in severe cases doctors will have nothing to do, just perform amputation, and it will have very serious consequences. For current lecture, that's all. See you in other lectures. Bye bye.